These are the lectures for Unit 4 on cellular structure. One thing that I do not like at all about teaching biology online is that we don't get to use actual microscopes, and I feel like that's something that is missing from the online biology. We do have a good microscope activity that's posted in if you scroll all the way down to the end of Unit 4 on Moodle, it's posted in the videos and articles resource that I put. Um, I'm not sure which one it is, but it says it on there, you know. But anyway, we will talk about microscopes, at least what they're good for, which types we use for which reasons and that kind of thing. But biology is really dependent on microscopes, particularly when we're studying microorganisms. But we even use microscopes to study tissues and that kind of thing. So a microscope is an instrument that magnifies an object. I'm pretty sure you knew that already. What we use in biology lab is called a light microscope. The highest that a light microscope can magnify an object is 1,000 times. But in the 1930s, the electron microscope was invented, which can magnify an object, I'm not sure how many times. It has a much higher magnification, higher resolution, and more detail. So what you're looking at in picture A is from a light microscope, but picture B is an electron microscope that is showing you, I mean, I can recognize this is a bacterial cell, um, and trust me, it's way more detailed than what a light microscope would show. Would be able on a light microscope, would be able to see this, maybe not even that big, you know. So I'd be able to tell that those bacterial cells were kind of oval shaped or rod shaped, but I wouldn't be able to see all that detail. So electron microscopes are used for, particularly if we want to see the inside of a cell, the structures inside the cell, which we're going to learn are called organelles. And if we want to see viruses, we don't have a choice but to use an electron microscope. All right, so when studying cells, one thing to know is that most cells are too small to see without the aid of a microscope. Even human cells, we need a microscope to see them. Within cells, there are numerous structures that also need microscopes. And in fact, if we're looking at structures within a cell, we almost always need an electron microscope. Now, at Wilson Community College, we do not have an electron microscope, which is not that uncommon because they're really expensive and you're only going to find them in places that need them. And we don't have that much of a use for electron microscopes at our college. We teach some of the beginning college level biology classes, but we don't go into, we don't teach the higher level biology classes. And that's where you really need electron microscopes. In eukaryotic cells, the numerous organelles means little organs, the organelles have specialized structures and functions, and you can only see those organelles with an electron microscope. And let me remind you, a eukaryotic cell means that it has a true nucleus and membrane-bound organelles. And part of what we do in this chapter is we learn about those organelles. We learn their names and we learn their functions and a little bit about their structure. Now, it's important for you to understand this is just representing a typical animal cell. What you're seeing here in this illustration is a typical cell, but our cells don't all look exactly the same. They're not all the same shape. And I mean, like for example, a nerve cell is very elongated. Your skeletal muscle cells are very elongated. So nerve cells kind of look like this. They have a cell body, but then they have these extensions that receive nerve impulses, and then they have extensions that carry nerve impulses away to some target. Um, and muscle cells, like I say, are elongated. So fat cells are big, fat, um, filled with lipids and fats. So cells are their structure determines their function. But this is just a typical cell so that we can study the organelles that you would find in a typical eukaryotic animal cell. First, we'll look at the nucleus. This structure, this entire structure is the nucleus. And inside the nucleus, you can see some dark purple thread-like structures, which are your chromatin. And chromatin is DNA plus proteins. So basically, the DNA inside the nucleus of your cells is in a form called chromatin. And then the nucleus is surrounded by a nuclear envelope, which is a membrane. It's actually a double membrane, and it has pores that allow material to move in and out. And then the structure you see inside of the nucleus is called the nucleolus, and it is inside the nucleus. So they're two terms that are easy to get confused. It is the region where ribosomes are formed. So let's find ribosomes. Ribosomes are not on this list, and the reason they're not on this list is they're not technically organelles. Um, all cells actually have ribosomes, even prokaryotic cells. But ribosomes are these little red dots. You see, I don't see ribosome anywhere. Okay, so a ribosome is just these little red dots. You can see them floating around free, and you can see them attached to the rough endoplasmic reticulum. It says here rough endoplasmic reticulum is associated with ribosomes, and that it makes secretory and membrane proteins. So ribosomes make proteins. They're not organelles because even prokaryotic cells have them, and they're not surrounded by a membrane. An organelle has to be surrounded by a membrane. But ribosomes do synthesize proteins. So it's important to know them as well. And it is the nucleolus inside the nucleus that makes the ribosomes. Okay? And then we have organelles called peroxisomes. Peroxisomes help with detoxifying our cells from different types of toxins. And peroxisomes can break down amino acids and fatty acids. So they're good for that. We have another organelle called a lysosome that can digest most any chemical and or digest most any chemical from food and break it down into waste. And lysosomes can also be used to help digest and break down bacteria in a white blood cell, for example. We've got the plasma membrane, which is the structure that encloses the cell, and it allows certain materials in and certain materials out. The cytoplasm is actually defined as anything between the plasma membrane and the nuclear envelope. So the cytoplasm includes the fluid-filled area, and it includes all of the organelles that you see. Anything that you see between the plasma membrane and the nuclear envelope is going to be the cytoplasm. But then we also have a term called the cytosol, which is just the fluid. And we have that on a slide in a minute, so I'm going to leave it for now. The word vesicle is not on here, but vacuole is. A vacuole is a large storage organelle And then a vesicle is a smaller storage organelle that's used more for transport than storage. So vacuole and vesicle both start with V. Just remember vacuoles are larger, and they don't have a picture of a vesicle on this one. Technically, a lysosome and a peroxisome are vesicles, but they're specialized types of vesicles. They're vesicles that contain certain enzymes inside, and they have a certain function. Then you have in the endoplasmic reticulum, by the way, the rough is here. It's called rough because it contains ribosomes attached to its surface. And so the rough endoplasmic reticulum is attached directly to the nuclear envelope. And then the smooth endoplasmic reticulum does not contain ribosomes. And instead of making proteins, smooth ER makes lipids. So we shorten endoplasmic reticulum and call it just ER most of the time. We will discuss the cytoskeleton of cells. It's not made of bones, but it does help to organize and give the cells some structure. There are three types of fibers that you find in the cytoskeleton. You have microtubules, which are the smallest. I'm sorry. Oh my goodness. Microtubules, which are the largest. I saw micro and I just forgot for a minute. Microtubules are the thickest, okay? And microtubules are found within a structure called the centrosome, which we will talk about a little bit more. It's important in cell division when the cell divides. Intermediate filaments are medium-sized filaments in between the microtubules and the microfilaments, and their purpose is to hold organelles in place so they don't move around so much. The microfilaments are the thinnest filaments, and you can see them. They're the green ones right here. They sort of arrange themselves um, right up against the plasma membrane, and they form the cellular cortex. Cortex just means the outer part of the cell. So they arrange themselves right up next to the plasma membrane and give the cells a little bit more support. Now we're going to have slides on all of these terms, but I do want to show you what is different in a plant cell versus an animal cell. The animal cell did not have a cell wall, only a plasma or cell membrane, only a plasma membrane. The plant cell has a plasma membrane, but it also has a cell wall outside of that plasma membrane to give it extra support and maintain its shape. A plant cell also has a very large central vacuole. We see a vacuole in the animal cell that's much smaller and less important. But in a plant cell, this is the vacuole that stores water um, and sometimes other components. A plant cell does not have a lysosome, so anything that needs to be broken down or digested happens in the central vacuole. Plant cells have a cytoskeleton just like animal cells, but plant cells have chloroplasts to carry out photosynthesis, and plastids, which are vesicles that store pigments. Um, Everything else, I believe, well, the plasmodesmata, you wouldn't have that in animal cells because they are channels between plant cells that connect one plant cell to another. But other than that, everything is the same. So in a plant cell, you have three structures that an animal cell doesn't, and that is a cell wall, a large central vacuole, and chloroplast. And animal cells have two structures that plant cells do not, and they are lysosomes and centrioles. Centriole is not on the slide, but we will talk about centrioles in just a minute. On this slide, it's just talking about cytoplasm and cytosol. Inside of a cell, the cytoplasm is everything between the plasma membrane and the nuclear envelope. So it includes organelles. and the cytosol is simply the